So testosterone is an anabolic androgenic steroid or hormone. So what it is it's got two components. It's androgenic, which means it's making a man. Andro meaning male, genic meaning to make or to start. And so it has this androgenic effect, and then it's got an anabolic effect. So these are anabolic androgenic steroids, okay? And so that means that testosterone is a steroid hormone. It's androgenic in its role of the dihydrotestosterone receptor. And so the dihydrotestosterone receptor is a receptor that basically binds to dihydrotestosterone. So in the process of making, this receptor is going to drive masculine biological shapes and forms. So in order for you to look like and have the secondary sex characteristics of a man, what you need to be able to produce dihydrotestosterone, and that needs to bind to the receptor. So there are a small group of families in the world that actually have this really unique component where about 50% of all of the offspring of those families are born as girls, and then at about 13 years of age, half of the girls turn into boys. And the idea there is that they lack the enzyme that converts testosterone to dihydrotestosterone. So dihydrotestosterone it binds to its receptor much better than testosterone does. So when there's low levels of testosterone before puberty, then the dihydrotestosterone receptor doesn't think that this is a boy, because there's no conversion of testosterone to dihydrotestosterone, so there's nothing binding to the DHT receptor. When that individual goes through puberty, now testosterone starts being made at a really, really high level, because even though the testes haven't dropped, they have developed. So now what happens is testosterone goes up really, really high, and now it can actually bind to the dihydrotestosterone receptor, and you can develop the secondary sex characteristics of a male. We see the same thing called male pseudohermaphroditism in women who start to take anabolic steroids or anabolic androgenic steroids. So there's a number of different groups where women will start taking high levels of testosterone. The testosterone will bind to the DHT receptor, and they'll get secondary sex characteristics of a man. It won't be a complete conversion like in the individuals who lack the enzyme to make dihydrotestosterone, but you'll get this pseudohermaphroditism. You'll start to develop certain traits of a man. They're anabolic, are mediated by the androgen receptor through both transcriptional and post-transcriptional mechanisms. And so the idea is that these things are going to increase muscle mass. So when testosterone is high, you get an increase in muscle protein synthesis and a decrease in muscle protein breakdown. Remember back to our bank account experiment, we're making more money, we're spending less money, our bank account's getting bigger. The other thing that they do that regulate muscle mass is they can increase satellite cells. And these are the stem cells that are resident within a muscle that by increasing satellite cells, you have the capacity to generate or to regenerate muscle faster. So here's data on testosterone and protein synthesis rates. So this is the fractional synthetic rate when you take this oxandrolone, which is a synthetic testosterone, and you can see that from control to 3 to 5 days of oxandrolone, you can see that there's an increase in muscle protein synthesis. This is total muscle protein synthesis. We don't know whether this is myofibrillar, so the contractile muscle protein synthesis, which is going to cause a bigger muscle. After resistance exercise, we see that the increase in muscle fractional synthetic rate is a little bit bigger, but again, this is total, not fractional to say that it's myofibrillar. This is total proteins synthesized within the muscle go up from either testosterone or resistance exercise. Okay, so there is this increase in protein synthesis that happens with testosterone, but just as much as protein synthesis goes up, it's going up about 20% here. What you can see is, or almost 30%, with 5 days of testosterone treatment or oxandrolone treatment. Over that same time period, you're actually seeing a decrease in protein degradation. So you're going from 83 to 69 units. And so that's a decrease of 17% decrease in protein degradation. And the way that the protein degradation is regulated, or sorry, let's first talk about synthesis. The way that we think testosterone is regulating protein synthesis, and this is done now in cell culture. So this hasn't been done in humans yet to show whether this is true in humans. But when you put in testosterone, you see an increase in the protein within the well in cell culture on muscle cells. When you treat with rapamycin, you can decrease, first of all, you decrease the total protein per well. And then when you treat with rapamycin and testosterone, rapamycin again is blocking mTOR. What you can see is that from rapamycin to rapamycin plus testosterone, you've lost the testosterone effect. And so what this is suggesting is that testosterone can increase protein synthesis in muscle cells, and that this is mediated in a process that's regulated by rapamycin, which suggests that it's dependent on mTOR complex 1 or a combination of mTOR complex 1 and 2. As far as the degradation rate, just like testosterone, when it's at really high levels, can wash over into the dihydrotestosterone receptor, another receptor that it can bind to is the cortisol receptor. And so this is the first experiment done in 1975 by Mayer and Rosen, where they took different forms of testosterone and they looked to see how much whether it could compete with cortisol for binding to the glucocorticoid receptor. And remember, the glucocorticoid receptor, glucocorticoids or things like cortisol, they're designed, one of the things that they do is they increase the rate of protein degradation in muscle. So they break down proteins from muscle so that those proteins can be converted into sugar, because cortisol is a stress hormone that's released when there's a need for sugar or for glucose. And so one of the ways that you can get glucose when you're fasting is you can break down muscle protein, and that can then be converted into sugars. Okay, so what we see is that when you add any of the different testosterone derivatives, you can compete out a synthetic cortisol. So you can compete out, so you can make it so that you decrease the binding of dexamethasone when there's testosterone. You also compete out cortisol. So 50% of the cortisol no longer is bound to the glucocorticoid receptor when the experiment is done with testosterone present versus when there was no testosterone present. And you can say that the different analogs of testosterone have different effects on the ability to bind of cortisol to bind its receptor. So what this is saying is that one of the ways that testosterone can decrease protein degradation is by binding to the glucocorticoid receptor. When it binds to the glucocorticoid receptor, it prevents muscle protein degradation. Just so that we can see that this has got ramifications in men versus women. In men that have 10 times higher testosterone level, if you look at how much cortisol is bound to the glucocorticoid receptor, you can see that having more testosterone means that there's less cortisol bound to the glucocorticoid receptor. In females, you have about 3 moles of cortisol per mg of glucocorticoid protein, of muscle protein, bound to this glucocorticoid receptor. And that means it's almost twice what the males see. And so what this is saying is that in females, cortisol or stress hormones actually have a greater negative effect on muscle mass. And we know that from other data as well, that females are more sensitive to the stress hormone cortisol. They'll lose more muscle mass under stress than men do, in part because men have higher testosterone levels, which can compete against cortisol for binding to the glucocorticoid receptor. All right, so that means that for women, one of the reasons that women tend to have smaller muscle mass than men is, again, going back to the analogy of the bank account, women are spending more of their bank account because there's more degradation, there's more going out of their bank account. And as a result, the amount that's in the total sum of what's in the bank account, because they are spending more essentially, their total amount is less than the males who are spending less. Okay, so if they're both synthesizing at the same rate, but the females are degrading at a higher rate, the males should end up with a bigger bank account or a bigger muscle mass.